Okay, it's 11:01. I think we've waited long enough. Thank you so much, everyone, for your patience. And yeah, I think we're ready to get rolling here. So thank you for joining. My name is Anthony. I'm the Director of International Business Development here at Rapid Novor, and I'm happy to have you all here today. We're going to be talking about anti-drug antibody assays, and in particular, the use of repab's polyclonal antibody sequencing technology in order to complement those existing assays in their development. As a quick overview, for those of you who are unacquainted with us, I'm going to let you know who we are. We're going to go through a few different antibody discovery workflows. We're going to examine some anti-drug antibody assay workflows, and we're also going to consider the repab case studies and successful projects that we've completed thus far, and some future directions that we're taking the technology. So, who are we? Well, Rapid Novor has been in the business of protein sequencing for quite some time, but we've also been in the business of proteomics and software development for even longer, nearly 20 years. But in the past 6 years, Rapid Novor has been able to perfect the art of protein sequencing in monoclonal antibodies, but also really driving the innovation behind the polyclonal antibody sequencing platforms that exist. So we've helped over 400 different clients worldwide, many of the top pharmaceutical companies by revenue, and we've successfully completed well over 3,200 monoclonal antibody sequencing projects. So most of those projects were completed, like I said, with the REMAB, monoclonal antibody sequencing platform. But of course, today we're going to be focusing a little bit more on repab. For polyclonal antibody sequencing. Of course, that's not all that we're up to. For those of you that are familiar with our company and familiar with our team, indeed, we are working on other technologies in the background, in particular, Novor, which is useful at decoding the immune response by quantifying and examining many different clones that are present in a given immune response. In addition to MatchMap, which is useful for confirming sequence similarity between, let's say, monoclonal antibody A and monoclonal antibody B, which are just different batches of an antibody which can be directed against the same target. We would be able to perform a series of experiments which will allow us to confirm the sequence similarity. But that's enough of the introduction. Let us move on to the topic of today. So obviously, Rapid Novor has been in the business of sequencing proteins. And the way that I like to think of this is that we're turning the central dogma of biology on its head. What do I mean by that? Well, the dogma posits that you start off with DNA, which is transcribed into RNA and then translated into a protein of interest. The dogma states that this is the way that it occurs in nature and that it only occurs in this direction. I like to think that Rapid Novor is also a part of nature, and that in fact we start with a protein of interest and then derive an amino acid sequence, which allows us to generate DNA, and then go back and begin this process from inception. So in this way, I like to believe that we are turning the central dogma of biology on its head by sequencing these proteins directly. And of course, I think that this allows us to evolve antibody discovery. Our team isn't limited to any particular species in terms of its sequencing, because fundamentally we're sequencing peptides, and those peptides are present in everything, right? So naturally, we've successfully completed monoclonal sequencing projects in just about every species you see here. And in fact, we've also completed now a number of polyclonal antibody sequencing projects in a number of species here as well. In particular, rabbits, goats recently, canines we've completed as well, and we're examining mice and humans and a few other options as well. Those projects are underway, and as we continue to develop them, we're happy to share more results. I thought that this might be of particular use to some of us that are attending today, because I think that one of the main advantages of being agnostic towards a species is that you can derive monoclonal antibodies for a specific species of interest. That could enable particularly useful insights into an animal model of interest. For example, if you're working in a canine situation, you may want to generate monoclonal antibodies against your target of interest, which are found in your canine, as opposed to working with a murine model, which may not recapitulate the context of that canine in vivo scenario. And this way we can generate monoclonal antibodies directly from this animal, and in the same way other animals as well. So conventional antibody discovery technologies, of course, start off with the hybridoma. wherein we're taking our immunized animal of interest, usually a mouse, extracting the spleen cells, the B cells in particular, fusing them with myeloma, doing your fusions, culturing those cells, and then harvesting your monoclonal antibody of interest. Of course, we would have to include display libraries into this context as well. Working with phage or yeast display has become quite popular, in particular in the past few years. But of course, phage display offers us a conventional way to perform antibody discovery as well. And of course, there's B-cell cloning becoming very popular these days with innovations such as the Berkeley Lights Beacon, the 10x genomics platforms, and of course, conventional flow cytometry methods from which we can sequester B cells of interest. I think that, I'm not going to come up here and tell you that our technology is necessarily better or worse, and I don't think that that's true of like any of these technologies. I think that everything has its own particular context and they're all complementary. And it's very important to remember that. Because I think you're going to get something different out of a hybridoma than you are out of a display library, than you are out of a B cell cloning library. And for that reason, it's important to consider, the diversity and the individual characteristics of each one of those options. You might have the ability to run a hybridoma program, and that's great, and that might work in that context, but there's other contexts in which it doesn't necessarily perform the best. And I think that, in any antibody discovery consideration, it's always important to just, think about your specific context as you develop your antibodies. But of course, I didn't want to spend too much time talking about monoclonal antibodies because we're here to talk a little bit more about polyclonal antibodies, particularly anti-drug antibody assays. So of course, I thought it might be useful to kind of go through a couple of these workflows and kind of examine where we might fit in our technology for the purposes of an ADA assay development. So with a conventional polyclonal antibody development program, you're going to inject your antigen of interest into your animal, perhaps a rabbit. You're going to pull out the bleed and perhaps pool, multiple bleeds from multiple animals together into a large batch, and then you're going to purify all of that into a purified polyclonal antibody, which is then fit for purpose. I think that this process is somewhat straightforward as compared to the monoclonal antibody antibody discovery platform, wherein you're taking your antigen, harvesting B cells, fusing them with a myeloma, and performing clone selection in order to generate your monoclonal antibody of interest. I think that, depending on who you ask, it could be the case that this latter process is more time-consuming, more resource-intensive, and takes a different skill set in order to generate those monoclonal antibodies. There are some advantages in doing so because you have a singular monoclonal antibody directed against your target of interest, and that can be strictly defined and characterized. But I think that generally, in terms of producing these antibodies, one might argue that the polyclonals are a bit easier to produce efficaciously against your target. But of course, once you have a monoclonal antibody and you're happy to work with it, you might want to use it forever. You might be really keen to say, I like this antibody a lot, I want to continue working with it. And rightfully so, you might want to immortalize it. So most people will go through the process of gene sequencing to derive the amino acid sequence, and then faithfully recapitulate that initial monoclonal antibody with the recombinant expression technologies. While that works great most of the time, there are instances in which it doesn't. Again, this is where the nature of complementary protein sequencing technologies can fit in. And this is what we've been able to achieve with this particular scenario. You might have a hybridoma that maybe not be performing particularly well. It might be difficult to clone that original hybridoma, and for that reason, using the protein itself might be of great utility in that context. Extending this idea a bit further, taking into consideration the polyclonal antibody development program, this is where we really feel that REPAP can offer an additional level of insight into these samples. So again, you're starting off with your polyclonal antibody, but being able to recapitulate the dominant monoclonals that are present and expressed in great abundance in that polyclonal will allow you to indefinitely express a panel of monoclonal antibodies, which again, faithfully recapitulate that original polyclonal antibody. And so in this way, we're able to kind of take the good parts of the PAB. So they're easy to produce. There's many existing products in the market. They naturally have a high affinity, perhaps in part due to their broad biophysical diversity. And we're able to mitigate some of these downsides, particularly the batch-to-batch variation, the limited supply, challenges in upscale production and the binding not being strictly defined, and if for that reason difficult to characterize. And I'll touch more on that later. Whereas in the monoclonals, you can do recombinant antibodies. That's great. You have an indefinite access to that monoclonal antibody with a digital sequence. It's very standardized, in terms of its production, and they're obviously easier to characterize because you have one antibody against one target. But of course, like I said earlier, they could be harder to produce initially successfully, depending on your target. And like I said, some targets just might pose a greater challenge than others. So effectively with RePab, we're able to take the good and discard the bad, and really effectively offer a novel way to discover these antibodies fit for purpose. So I'm going to stop here for a minute and initiate a quick poll. Let us launch. So what was the longest it has ever taken you to develop an anti-drug antibody assay? I'll be sharing these results with you all here. I'll leave it open for about a minute or two as I check on the questions. I'll be saving the questions for the end, folks, so I will get back to you. Wahala, I see you. Last one here. Very good. We have quite a few answers. Okay, and I will stop there. Looks like most of you can actually generate these ADAs in about 1 to 3 months, which is good to hear. But in some cases, it looks like it can push upwards of 18 months, which I'm sure can delay timelines going into clinical trials and other kinds of milestones that are probably pretty critical to any therapeutic development program. But yeah, we'll be. Oh, I should share these results. My bad. Here we are. Yes, now you can all see. But yes, it's good to see that there's some variation in this. Okay, excellent. So I will stop sharing results. Very good, and we will move on. Okay, thanks so much, everyone. And we'll be happy to share those results with you afterwards as well, in the follow-up emails. Okay. Now that we've taken a quick break, I wanted to quickly kind of jump back into the anti-drug antibody specific polyclonals. I kind of talk about some of the drawbacks that we've observed from some people that we've spoken to. In particular, you're pulling a lot of antibodies. You're pulling a lot of samples from many different animals, and that can be, challenging for some reasons. In particular, you can have a lot of challenges in terms of characterizing those antibodies. You don't know specifically where they bind, and depending on where you are, particularly in the EU, using a lot of different animals in your therapeutic development pipelines may pose a problem. You might have to justify the decision. It might preclude some of your options, and for that reason, it's kind of a drawback. You naturally have a very finite supply of these samples. Like once you've gone through the immunization process and generated your PAB, pulled all your samples together, that's it. You might have a few other antibody animals from which you can continue to draw from, but ultimately, there's going to be some variation within those samples, and for that reason, there might be some challenges with respect to validating the results that you got in the initial batch. Because it's polyclonal, your binding is not strictly defined, and I'm going to really point out why that's more of a problem in the next few slides here as well. And like I said earlier, it might be the wrong species. I hope you like this picture. This little dog, is not fooling anyone. It's not a mouse, but certainly, in your context of interest as well, it may not be sufficient to engineer an antibody into a particular species of interest. It may actually be required, or at least maybe more beneficial to start off with an antibody that is fit directly from the species of interest. Okay, so as a quick review, let's just make sure we're talking about the same thing. So we're talking about anti-drug antibodies, and I'm particularly anti-drug antibody assays, and I'm particularly talking about the mesoscale discovery platform, wherein we immobilize the drug of interest into a solid phase against a solid phase, and then we flow over the polyclonal antibodies from the patient of interest, and then one Fab arm will bind against the drug, and then that is immobilized, and then the other Fab will be able to bind against a labeled version of the drug, letting us know, in what proportion that drug is bound to the anti-drug antibodies present. This is a nice scenario because it's a nice picture and it's one antibody against one drug, and that's nice to think of. But obviously, a polyclonal antibody can be very different. You're going to have a lot of different antibodies that are present in a real life sample, and it's challenging to strictly define that interaction on its own, right? Like you need something to compare it against. You need a control antibody to serve as a reference point for the serum that you're flowing over from the patient. And that's important because if you don't have a reference point or a solid reference against, you have these antibodies binding against many different areas on the drug of interest, but you don't necessarily know where or of what type or what level of affinity they are. And for that reason, it could be very challenging. It can be more challenging to characterize this as compared to like a known reference panel. And so this is really where we see antibody discovery offering a lot of assistance from the polyclonal perspective, is that you can come in with a strictly defined monoclonal antibody reagent, and then you know exactly where it binds, you know exactly what the isotype is, you know exactly what the affinity is, and then from there you can have a really nuanced context from which you can compare this reference panel against the polyclonal that you're observing from the patient themselves. So to offer some more context to this and to kind of make it a little more concrete, I thought we should look at an example. Of particular interest is factor VIII. Factor VIII is an interesting protein. It's essential for blood clotting, and it is encoded by the factor VIII gene. Defects in this gene will result in hemophilia A. And Genentech was the first company to really generate a recombinant, which served well because prior to that we were just isolating it from plasma. But really using the recombinant form, proved quite efficacious for these patients. However, even today, we still use this and prescribe it quite often. But even today, though, there is significant levels of immunogenicity in the recombinant human version and also in the recombinant porcine version. Actually, 25 to 30% of people who take the recombinant human version of the drug will actually generate an anti-drug antibody response, which can ultimately be important to consider when considering the clinical implementation of this drug or its analogs. And so for this reason, it's quite critical to have a highly sensitive and high fidelity assay from which we can make informed decisions for clinical practice. And so naturally, as I was saying earlier, it can be very important to define strictly where the antibodies are binding. In particular with the factor VIII case, A2 and C2 are these two domains that I've labeled here. And these are really important to remember because they are neutralizing antibodies, neutralizing regions from which if an anti-drug antibody response is binding to, that'll neutralize the drug and it'll effectively render it useless. And that can be very problematic, but also very important to know as we develop these assays. And extending on that further, the C1 domain is of keen interest as well because it is non-neutralizing and still potentially pathogenic. Indeed, because it is non-neutralizing, it'll bind to the C1 domain, which will actually make it quite a bit more challenging to clear the factor VIII protein from the patient of interest. So, with this example, with this context, we can see that it's quite important to understand not only, whether or not you have antibodies binding against the protein of interest, but specifically where and why that impacts the clinical practice. And beyond that, the isotype can also have a great importance as well. IgMs, could indicate an early response to the drug of interest, and that may be useful because maybe this is not a robust and developed antibody response at this time. You just have the IgMs present, but once you start seeing additional antibodies present, like your IgGs and your IgAs and your IgEs, this can offer some different insights as well. And in fact, other instances in which people have derived panels, they've really tried to offer isotype controls in their panels to really suss out the exact binding characteristics of these antibodies. In particular, with factor VIII, they found that some researchers found that with IgG4 maps, they were found exclusively in inhibitor patients. So it can be quite useful to have some kind of assessment for just those IgG4s in that context to understand, okay, are these antibodies inhibiting? And for that reason, again, guiding clinical practice. And so I think that, with these examples and with this context, now we can see, well, we can answer this question, hopefully. It doesn't take too much. Why do we need reference panels? I think this is a really good reason, right? The usage of these assays can vary greatly with no standardized positive controls. In fact, some researchers have also studied, again, this particular case and found that there's a lot of variance between labs that are conducting the anti-drug antibody assays for factor VIII, such that the variance is actually like 40 to 60% across 15 different European labs. So part of that was because of like the instrumentation, but also, the authors attribute a significant part of that to the lack of standardized positive controls. Because these antibodies that you're using, you might not have access to patient ADA responses that may be challenging for you to get your hands on. And if you lack a robust panel of antibodies which can faithfully act as positive controls, you may have a lot of variance, not only in terms of how you run your assay, but in terms of the different assays that you run, like SPR versus BLI versus ELISA versus flow cytometry. The antibodies might perform differently in each one of those assays, and for that reason, you might have some variance. So offering a really robust panel of antibodies can faithfully act as positive controls is quite critical. So the NIBSC from in the UK agrees. They've actually gone ahead and done this, and in tandem with the WHO for, well, in particular EPO. They found great success in doing this. So they actually partnered with Amgen, so the WHO, NIBSC, and Amgen partnered together to make an ADA panel which contain 9 different monoclonal antibodies against different epitopes, so different isotypes and different affinities, to offer a range of monoclonal antibodies which could recapitulate the patient ADA response. These are human mAbs, but they weren't derived from patients. And so I like to say that going directly to the patient themselves may offer a greater clinical significance, but I'll get to that. I'll get to that in a minute. Coming back to factor VIII, they've gone ahead and done this as well. So they've actually generated a panel of antibodies, which again act as those faithful positive controls, and you can get them. You can buy them directly from the NIBSC, and they offer that, and in that way we can standardize this approach. We can standardize these assays and strictly define their binding characteristics with respect to their assays and their particular context. So in terms of like how a project looks, or in terms of how this works, so we start off with your patient or animal of interest, which has been immunized with your drug. And then from there, we can pull out the serum, and then we can actually deconvolute that signal a bit further by using, for example, positive control reagents if it's a really big protein, or if you have a surrogate peptide that can act as like an epitope of interest. This can also work, but I'll be moving forward with this positive selection reagent kind of concept, wherein you have an FC fusion protein attached to a domain of interest. And then from there, you can take a column and you can immobilize your FC fusion protein in that column, which will capture the antibodies you flow through that are specific to that domain, and then allowing you to flow through the remaining antibodies, but then, like I said, capturing the domain-specific antibody, polyclonal antibody of interest. And that's good because from there we can actually work with it and we can do sequencing. So this is a quick snapshot of our proteomics sequencing workflow. I should say proteogenomics because we can use genomics as well to complement this workflow. But in essence, we're starting with the animal of interest. We're purifying that serum. We're putting it through our serum proteomics workflow, generating prospective heavy and light chain sequences, which you can then pair into their cognate molecule, and then have a full-length IgG, which can then be expressed as a recombinant and then faithfully recapitulate the antibodies that were present in that sample of interest, which is hopefully coming from a patient, but of course it could also be coming from like an animal, like a rabbit, as they are quite conventionally used in ADA assay development. Of course, this is the proteomics workflow. If we have access to the PBMCs or any other such genomic, B-cell rich tissue, we would be happy to perform transcriptomic analysis and generate NGS reads in order to complement the proteomics workflow that is seen on the top arm. And so we can generate a really broad and diverse panel of monoclonal antibodies that are found within that polyclonal serum. And I'll be getting into a bit more detail on this in a moment. So yeah, just hold tight if you have questions. But of course, ask the questions in the panel if you're inclined to. So again, coming back to the original example, by starting off with
a strictly defined monoclonal antibody, we can offer a broad range of, we can offer a comprehensive panel which can faithfully recapitulate the binding activity of the polyclonals that you see coming out of patients in a number of different scenarios, whether they're binding against particularly neutralizing domains or non-neutralizing domains, or whether there's an isotype switching phenomenon that's driving some kind of other interaction. This is what having standardized controls really affords you, is a reliable comparative. And of course, these are my opinions about this kind of topic, but I feel, and I think others might agree, is that this is the most faithful representation of the patient anti-drug antibody response, because you are deriving those antibodies directly from patients when you have access to that material. I think that this improves accessibility quite a lot as well, and as a result, it reduces the variability with respect to these assays when they're conducted, wherever. When you can just go to the NIBSC or some preferred vendor and buy a panel of antibodies, you have access to those pretty regularly, as opposed to needing to somehow source these anti-drug antibody responses directly from patients to act as your positive controls. You might not have access to them, and for that reason, having access to a panel that's already been described and sold by a vendor could inherently increase the accessibility. We're working with proteins, not genes, which is great, because if you do a single cell sequencing approach, of course, you can generate antibodies. But I think that, my opinion here is that because we're working with the protein itself, we are observing the most abundant proteins that are present in that patient. And naturally, I think that more faithfully recapitulates what's going on than relying on the RNA transcripts or however, whatever genetic material you're using to perform that single cell sequencing. I think that this also offers us more nuance, so we can track parameters outside of just the binding affinities for a target epitope. Like I said earlier, you can generate a lot of different monoclonal antibodies against these targets, and that's not the problem. But sometimes it's a matter of understanding, is the anti-drug antibody response itself going to cause problems? Is it going to be immunogenic itself? Is it going to aggregate? Are there other problems that we need to consider with respect to these monoclonal antibodies that are being generated? Are they potential neoantigens? This is something to consider as well. Again, this is my opinion, but this is why I feel that working with patients, ADAs are the best way to kind of move that forward. So I will stop here again for a quick poll and ask, how do you assess immunogenicity in your pipelines? I'll leave this open for about a minute, and then we'll come back to the rest of the presentation. I find this question interesting because we've been speaking with some people in the industry as well, and some people's opinion is that there's only one way to do this, immunogenicity assessment, and it is ADA assays. But if I totally understand that some people might have a different take on this, like cellular-based assays in order to assess not only the antibody response but also the neutrophil and macrophage and other cellular responses that can occur in response to a given drug. Okay, I'll end the polling there. Share the results. Looks like most people are doing ADAs though, which is, I guess, not surprising, and I'm happy to see that. But of course, there are other methods, and I think that generally speaking, other methods are becoming available as we continue this, yeah, in the world today. Okay, so stop share and resume. So from here, we kind of have an opinion about these assays and their development. What can we do about it? So for those of you who are, again, unacquainted with us, I'm going to walk you quickly through our monoclonal antibody sequencing workflow, because it's very similar to the polyclonal antibody sequencing workflow, and then I'll get more into that as well. So essentially, the way that we do these sequencing workflows is we start off with a sample of interest. We purify out the antibodies. We do a multi-enzyme digest. We then put that digested sample through the LC-MS/MS workflow, wherein we can generate spectra, and those spectra are then used to derive the amino acid sequence of a particular peptide. And once we have enough peptides, we can then assemble them into a full-length monoclonal antibody sequence. And it's really from there that we drew the inspiration to perform the repab workflow, which I showed a bit earlier, where we're doing the serum proteomics. In our case, we're typically working, when we're doing the serum proteomics, we're typically working with antigen-specific affinity purified material. And that way we have really narrowed the antibody response just against the specific target of interest. In most cases, that's a peptide. In some cases, it's a protein, which we can deconvolute using that kind of SC fusion domain that I mentioned earlier. And generally in a project, we do this in a few different phases. So we start off with the immunization. Once we have positive titers for the immunization program, we pull out that serum, purify it, and move it towards our screening. In our screening steps, we're able to assess those samples. And really what that means is, we're trying to assess sequenceability, for lack of a better word, in our own hands. That means that we're going to do a multi-enzyme digest and serum proteomics workflow, much like I showed you earlier, and just see what the data looks like. And then from there, we'll have a kind of binary output as to whether or not it can move forward, and we can go into deep sequencing. Once we've completed the deep sequencing, we have a prospective library of monoclonal antibody sequences, which you can then synthesize and then send off for analysis. Of course, phase 1 and phase 4, and as far as we're concerned, are optional. We have partners that work with us to do that stuff, but certainly, we're open to working with anyone else who's got that kind of material already in hand. And so that's the theory. How does it hold up? Well, in our case, we like to work with rabbits quite often, but not always. We like to work with rabbits, though, because, well, they perform quite well in IHC. They're big enough. That's also one of the challenges that we deal with in the polyclonal sequencing workflow, is that we need sufficient amount of material in order to run the analysis. Generally, we look for about 1 milligram of antigen-specific affinity purified IgG in order to faithfully run the pipeline, although we are always keen to improve our capabilities and minimize those material requirements in other contexts as well. But again, rabbits are nice because we have enough material. There's a robust NGS primer set available to us for B-cell repertoire sequencing, and for that reason, we really like working with them. They're also nice because they offer higher sensitivity and specificity compared to as compared to some other animals, and in particular against small targets like less than 20 mer peptides or PTM variants and haptens. I think rabbit models have been shown to be quite efficacious. Beyond that, they have a different ontogeny, and so they offer something complementary to, let's say, a different animal, like a mouse, right? You have different genes, you have a different kind of affinity maturation process, and so you're probably going to get something different out of that process than you would out of the traditional mouse hybridoma approach. And lastly, they have excellent stability due to an additional cysteine bridge within their variable regions. So there are some kind of straightforward applications within early stage research and development that we've been keen to explore, and so we did. So we converted, firstly, a PAB to a MAB or MAB cocktail. In our case, we used a peptide to immunize a rabbit, a couple of rabbits, and then from there, we generated some sequences. So I should say that this data comes from last year, and you're going to kind of see a progression in the next few slides from like where our projects were and where they are today. So this is the level of coverage that we achieved on these first couple of antibodies. In our experience using our proteomics workflow, we say that a minimum of 20x coverage is required to have a 100% accurate sequence for any given antibody chain, and subsequently, the full-length IgG. So in this case, you see we hit about 20x. 20x is where my cursor is. And we luckily achieved that level of confidence throughout this antibody pair for the heavy chain and the light chain, and this one as well. Of particular note is always CDRH3. This data looks really good. We're very happy to proceed, and in fact, it proceeded quite well. So we generated 3 prospective antibodies, 2 of which performed quite well as compared to the original polyclonal, which is PD030, seen here on ELISA. So R1 and R3 are some antibodies that we use regularly internally, and again, they perform quite well today as they did a year ago. SPR also confirms these results. So we ran SPR on these, and we see that the original polyclonal was a single-digit nanomolar binder, and so are the recombinants as well. And so naturally, we're quite happy with these results. They perform quite well in their intended application, and indeed, they have very high affinity. Next, we took on a project in a diagnostic context wherein the client wanted us to generate a monoclonal antibody against a peptide of interest, which was contained in a biofluid of interest in order to perform a relative quantitation of that peptide in that biofluid. So in a time crunch, they left us with only using proteomics, but in the end, they were quite happy. Indeed, by their own admission, this was a highly specific and high affinity panel of antibodies against this target, and they were indeed fit for purpose. So here is the result by ELISA for that original proteomics only panel of monoclonal antibodies. In this case, we did not have access to NGS data. We just didn't have it available to us at the time. And so we generated this proteomics only data set. And I should also point out, we're the only team who has ever successfully sequenced multiple monoclonal antibodies from a polyclonal serum and delivered them and fit for purpose in their intended application. I should say that eventually, though, we did obtain NGS data, and indeed, we leveraged it against this original data set in order to derive an additional panel of monoclonal antibodies. So in the end, we generated 12, all of which performed really quite well on an ELISA, and all of which performed really quite well in their intended immunoprecipitation application. Last case is that we worked with a goat just recently. We've just completed this project, so we can't confirm exactly the binding results just yet, but obviously I'm happy to share this with you all today because this was also done using only proteomics, and we took a goat polyclonal, single source goat polyclonal against a peptide of interest, and derived two dominant monoclonal antibodies, which have excellent levels of coverage. The coverage you see here actually goes a bit deeper than the images I'm showing you, but the screen resolution cut off in our case, and so we presented this data as it is. But as you can see, this is excellent level of data. We have coverage all the way down, all the way throughout the entire variable region of these antibodies, and we're quite excited to see how they perform in their intended applications. We will indeed be testing them across a number of different applications to see how well they perform. Okay, great. So what do we need in order to get started? Well, if you have polyclonal antibody that is derived from a single animal, I know this one can be a bit tricky sometimes, because if you have access to a polyclonal antibody, it was oftentimes pooled from multiple different animal sources. That's quite problematic for us. The heterogeneity problem is quite challenging for us to overcome. So naturally, we like to work with a single source polyclonal reagent if we can help it. So if you have such a reagent, that's great. Hopefully, if it's directed against a small enough target, we could be able to take it on or deconvolute that signal using some of the purification methods I proposed earlier. It is quite helpful for us to also have some blood with which we can work. 3 to 5 mils is usually more than enough, so that we can perform NGS sequencing against the PBMCs present in that blood sample. If you don't have the kinds of materials that I've outlined, you just have like an affinity purified material. You don't know if it's got multiple sources or not. Like, let's have a conversation about it is usually what I like to say. Or if you're working with a different species like sheep or mouse or hamster, we're open to having that conversation as well. Of course, we're keenly investigating mice. I know that there's a lot of interest in that, and we're trying to make it work. But yeah, the results are forthcoming, so I should say that. In terms of future developments, we're doing custom NGS development in order to enhance those capabilities in other species. We're always trying to improve our protein-only sequencing capabilities, and as I've been doing this presentation now for a few months, I can say that we've obviously been improving on that front pretty consistently now, as you've just seen between the first case that I showed you and the most recent goat polyclonal case. We've definitely made a lot of improvements in our protein-only sequencing capabilities. We're always trying to find more antigen-specific leads by doing NGS mining, taking a prospective NGS database and then complementing it with proteomics to investigate, further antigen-positive leads. And then not examining new methods for immunizations as well, trying to reduce the timelines as much as possible, looking at different species like camelids, and really trying to cut down the amount of time it takes to run a given project. So in summary, anti-drug antibodies can be sequenced directly from human patient serum. You can either using a combination of proteogenomics or protein only. RePap can deliver a library of antibody sequences useful in recapitulating that human antibody response to an antigen of interest. And of course, Novor is seeking royalty-free contracts for the use of this technology in the pharma and biotech industries. This is my contact information. You can capture it here. I'll probably also send you an email though, so you can always talk to me there. And again, thank you for your time. So I'm going to turn over to our questions. But please, if you have any questions, now's the time to ask, because I will be answering them for the next, well, 15, 20 minutes or so, as long as there are questions. And if there aren't, I'll just, I'll end it a bit early. Okay, so Wahala, you've asked, do you generally make canine antibodies as a day-to-day work? I mean, we've done it. So I would say that, it's not something that we've done regularly, on a consistent basis, but I'd say that we have done it. We are hopefully going to be doing more in the near future as well. And of course, we're keen to do more projects along this line. Yep. Ilya, nice to see you here. You're asking, what time do you need to do for PAB identification? I guess you're asking the timeline to run the entire sequencing workflow. Yeah, the sequencing workflow takes between 2 and 3 months. It just depends on the sample of interest and exactly how heterogeneous that sample is. But if you, that's just for the sequencing, I should say. That's usually broken up into like kind of the 2 middle phases I showed in my previous slide. We do about 4 to 6 weeks to do the initial screening, and then 4 to 6 weeks to do the complete sequencing of the chains and perform the pairing of the cognate pairs of those chains. So yeah, that's the timeline generally. Of course, you can add on the amount of time it takes to do an immunization and then synthesize those antibodies. Yep. Yep. How much blood is required to do the NGS workflow? Yeah, we ask for a few mils. 3 to 5 is, I think, more than enough. Realistically, 1 or 2 mils is also likely sufficient in our case to perform the NGS sequencing, because we're just doing bulk RNA-seq against the PBMCs that are present in the blood. We don't need that much blood. Of course, we're happy to receive samples from other B cell rich tissues, such as bone marrow or spleen or something like that as well. Yeah. Hi, Kuros. Thank you for your comment. Can I share today's recording? Yep. So we will be able to share the recording with everyone who attended here today. Actually, we're probably going to upload it to YouTube, so you can always check out our channel, like and subscribe. So that information will be available there as you need it, and if you want to share it with anyone else as well. Great. I got another question here asking about how we perform our pairing of our chains. So I should say that we're not using any genomic-based technologies. We're not doing like single-cell sequencing or like a 10x or a beacon-based kind of approach, and we're not labeling the genetic information in any way to derive that pairing. This is purely done through proteomics suite of experiments. Unfortunately, I can't be extremely transparent in terms of how we do that. Due to some developments on our end, we're trying to secure the intellectual property surrounding that technology. However, I can say that it has been empirically validated now through a number of different projects. We've been doing this now for several years. In the monoclonal antibody case, we're monoclonal antibody, quote unquote, is actually less monoclonal than you might expect. Oftentimes, actually, something like 3 to 5% of all of our protein sequencing cases we do observe additional chains. And I know that some other researchers, like Bradbury L, have certainly reported that up to 30% of a given hybridoma set may contain additional chains, whether they be additional light chains or additional heavy chains. And we've worked with those samples, and we've gone ahead and sequenced the chains that are present in those samples and then paired them correctly. Which we've done probably in a few dozen cases by now in the like I said, quote unquote monoclonal context. But now, after having performed several polyclonal antibody sequencing projects as well, I think that the results kind of empirically demonstrate our efficacy. If you didn't pair the chains correctly, I don't think you'd be seeing the level of binding activity that we are seeing in the samples that we have, in the sequences that we have delivered. So in that way, I can say that we have validated this approach to some extent. But yeah, that's basically what I can say about it.